the right way. In this opportunity, we are delighted to welcome you in today's event. Guest lecture Formasip Webinar 8 series with the theme Metabolomics Application in Anti-Obesity Compounds Discovery from Natural Products. My name is Beneficia Rosi Acha, and it is an awesome and prestigious change for me to be the master of ceremony in this event on Monday, 30th May 2022. And now let me welcome to our speaker and participants. Welcome to our main speaker, Honorable Professor Dr. Milen Georgiev, Head of Plant Cell Biotechnology Department Center of Plant System Biology and Biotechnology, Plovdiv, Bulgaria. Good morning, Professor Milen. Also, welcome to our moderator, Honorable Dr. Nancy Dewi Yuliana. Good afternoon, Dr. Nancy. And last but not least, welcome to all of the participants. Our highest appreciation and thank you for joining this event. Okay, ladies and gentlemen, before we start to our agenda for today, let us pray to the Almighty God based on our belief. Pray begin. Finished. And before we go to the next session, let us take a group photo first. Maybe from the documentation committee can help me. So here we have
there are 4 slides. So okay.
Okay, so we are going for the first slide. Please give your greatest smile on my count. 3, 2, and 1.
Next slide.
Next slide. 3, 2, 1. And the third slide, maybe.
Okay, next slide. Still many haven't opened.
Okay. Can we move for the last slide, maybe?
Okay.
And 3, 2, 1.
Done.
Thank you very much for the
Thank
cooperation.
you.
Okay, thank you. And now we will move to the next agenda. The next agenda is welcoming speech from Dr. Nancy Dewi Yuliana as a representative of Food Science and Technology Department. For Dr. Nancy, please.
Okay, thank you, Rosie, our great moderator today. Yeah. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Welcome to our honorable speaker, Professor Milen Georgiev, and also all the participants to this webinar. But actually, this is the guest lecture, of one of our course in Department of Food Science and Technology. Metabolomics Application in Food Science and Technology. So every year, Prof. Milen, we invite somebody, mostly from Rob's lab, including Kasif, and also now it's your turn, to give a guest lecture, because I know that you have a very, very excellent research experience in this field. We really appreciate, Prof. Milen, that I know that you are busy in your tight schedule. You still spare your time to be with us today. We really appreciate it. Thank you very much. And the head of Food Science Study Program, Professor Harsi Dewantari Sumaningrum, she sent her apology because she could not join us in this webinar. She has a class at the same time, but she sent her regard to you. Also, I would like to thank to all graduate students, yeah, that you can join us today. So please take the benefit from this guest lecture, yeah, as many as possible, because Prof. Milen, she has a bunch of research. Yeah, I will show you from his CV later, yeah, in this field. So please ask a questions, yeah, don't be hesitate. He will be happy to answer your questions, yeah. Okay, so I think without further ado, I know that the participants, they are very eager, yeah, to wait for Professor Milen's talk. So I return again to Mbak Rosi, yeah, please continue this session. Mbak Rosinya, where is she now?
I'm sorry, I have a signal trouble, Dr. Nancy.
Okay, that's good. That's okay. Okay, you're back.
Okay, so thank you very much for the speech, Dr. Nancy. And now we are moving forward to the main session that we are waiting for. From here on, I'm going to hand over the session to our moderator that will be lead also from Dr. Nancy Dewi Yuliana. And she is a lecturer in Department of Food Science and Technology. Some of her teaching courses is Chemistry and Biochemistry Laboratory, Food Analysis Laboratory, Food Regulation, and Halal Assurance System. And she has a research interest in metabolomics and multivariate data analysis and bioactive compounds identification. So without any further ado, Dr. Nancy, please, time is yours.
Okay, thank you again, Mbak Rosi. Yeah. Okay, so everyone, so before we start, yeah, I would like to read the CV from Prof. Milen. This is actually, I make it very short, yeah, otherwise he has a very, very long CV, and yeah. Okay, so this is the first page. Yeah, Professor Milen Georgiev, he is now a professor at Institute of Microbiology, Bulgarian Academy of Science, where he actually head of metabolomic labs, yeah, and also he is a head of Department of Plant Cell Biotechnology in the Center of Plant System Biology and Biotechnology in Bulgaria. He is actually an associate editor of many journals, yeah, but I only picked two of them, yeah, Medicine and Food and Chemical Toxicology, both belong to Elsevier. And he was a former Marie Curie Fellow at Leiden University, the Netherlands. Actually, I met him there when he pursued his postdoctoral degree, yeah, based on the Marie Curie Foundation. For your information, this Marie Curie Foundation is a very prestigious funding body, yeah. So if you receive a grant from this foundation, means you are an excellent scientist, yeah. And Prof. Milen got it twice, right? Yeah, because he was also a former Marie Curie Fellow before in Leiden, in Dresden University of Technology, Germany. And this is his current research of interest, biosynthesis of fine molecules and development of biotechnological tools for their sustainable mass production, and also in metabolomics and comprehensive metabolite profiling. I know he published a lot of papers about it, and molecular pharmacology, particularly focused on obesity, photoaging, and longevity. And this is the second page. Yeah, you can see his excellent corpus profile. She published 147 papers, yeah, and he has like more than 4,000 citations, and Scopus Edge Index is 32. I don't know, maybe until my retirement I will not reach that's very high score, yeah. Okay, so we are very lucky to have him today. Okay, so I think you are very eager, yeah, to wait for his talk. Without further ado, Prof. Milen, time and screen is yours, please.
Thank you, Nancy, for the kind introduction. And yeah, so good morning, everyone, or perhaps I should say good afternoon, as in Jakarta is already afternoon. So yeah, I'm sorry that I cannot speak in Indonesian or in Malay, but I assume most of the people in the audience don't speak Bulgarian, so I should try to make it in English. And first of all, I would like to thank Professor Nancy Yuliana for the kind invitation and introduction. So what I'm going to speak about today is a summary of what we do on metabolomics field, and I will give you some examples of the research which we do also on obesity. So we are interested in obesity for many reasons. In a short while, I will explain why. So the idea is just to give an overview of what we are trying to do. So if you have questions, you are welcome to ask some. Yeah, okay. So as Nancy mentioned, so my lab is lab of metabolomics primarily, which belongs to Bulgarian Academy of Sciences. And a couple of years ago, we established a center of excellence in my city, which we name Center of Plant Systems Biology and Biotechnology. So we managed to get a good support from different sources. So in European Union, we have framework programs which are supporting science, and we got a project from European Commission, supported Bulgarian government and the municipality of my city. So, and this is how our center looks like. We have a department of metabolomics, and here at the bottom you can see some of the papers which we published last year and recently, I should say recently, from my department. So later on, I will show you the website of the center so you can get back if you want some further information. Okay, so I'm primarily interested from plants. As you all know, we use plants for food and feed for many years, for thousands of years. But besides accumulating carbohydrates, fats, and sugars, plants accumulate a small portion of molecules which we call secondary metabolites. Actually, right now there is a tendency whether we should call them secondary metabolites or specialized metabolites, but in any case, many of them have shown beneficial properties for the human health. So here on the slide, I summarized some of the so-called blockbusters. And in the middle of the picture are paclitaxel, which whose structure you can see here, and it is used to treat at present different types of cancer. And I should say that the annual sales of paclitaxel and related taxans is worth of several billion US. And on the top is anti-malarial artemisinin, whose discovery was awarded with Nobel Prize a couple of years ago. So the Nobel Prize was awarded in physiology and medicine in 2015 to the Chinese scientist Yu Tu. So a couple of years ago in Vincenzo De Luca, in his science paper, actually it's not a couple of years ago, it's already 10 years ago, because the paper was published in 2012, Vincenzo De Luca estimated that the annual market of phytopharmaceuticals only in States is exceeding $25 billion US. So last year we were invited to publish a review on Nature of Products Drug Discovery for the Nature Reviews in Drug Discovery. So more recent evaluation of the field is available in this paper, and if you cannot get an access, please do not hesitate to get in touch and I will send you a copy of it. So this is one more example, which is coming from a very beautiful plant, Catharanthus roseus. Catharanthus is is used for a very long time to treat diabetes. However, something like 50 years ago, it was found that some dimeric indo-alkaloids like vincristine and vinblastine are having very, very good anti-cancer activities, and in particular with acute leukemia and breast cancer. At the same time, the content of these dimeric alkaloids in the leaves is 0.0003%, which in another word means that you need to process in excess of 500 kilo of plant material to obtain only 1 gram of vincristine. So as you can guess, processing or obtaining these compounds from natural harvest could create some really severe ecological issues. And if someone of you think that this is a little bit exaggerated, I put this example here, this picture, which is coming from European flora. This is a very beautiful plant which is called Leucoium aestivum, and it is a source of galantamine. Galantamine is an alkaloid which is nowadays used for treating Alzheimer's disease. So the over-harvest of this plant species in Bulgaria for many years resulted in severe problems with the natural habitats of this plant, and it was placed in Bulgarian Red Data Book, which means that you cannot anymore harvest commercially. And perhaps there are several also examples from the Indonesian flora, which is quite rich. So in other words, there is a necessity for acceleration. So we need to find a way to accelerate the process from the lead compound to the drugstore, to the pharmacy shelf. We need to find a way to do a quality control assessment. I will show you some examples later on how we use NMR for quality control assessments and to determine the mode of action. With the same time, we need to find a way for the sustainable production of these molecules. So those of you who are already working in the lab, perhaps you have some favorite plants due to whatever biological activity, but just imagine these beautiful plants, if we over-harvest them, if we overutilize them, what we will leave to the next generations. And just think for the plant species which you use, because according to the WHO, 80% of the human population, 80% of the human population still rely on plants for their primary healthcare needs. So just imagine the plant the medicinal plant which you most frequently use. If this plant was over-harvested in the past, now we wouldn't have possibility eventually to enjoy it and to use it for our healthcare needs. So sustainability and acceleration are the keywords, and I believe metabolomics and biotechnology are eventually able to give some solutions into this direction. So this is a little bit busy slide, but this is more or less the concept of work in my lab. As I mentioned, we are interested from plants, medicinal and aromatic plants, not only from Bulgarian flora and European in general, but also from all around the globe. So this one on the top is a plant which is called devil's claw, or Harpagophytum procumbens, and it grows only in South Africa and in particular in Kalahari desert regions. So what we do quite a lot of extraction, isolation, and structure elucidation. We are mostly using NMR, but in some particular cases, we also do some mass spectrometry. So in my lab, we have a bunch of in vitro as well as in vivo assays. We are interested in inflammation because inflammation is quite important. At certain level, inflammation is needed for our bodies to heal, but when it becomes chronic, it might be dangerous. So we are interested in information, and mostly we isolate pure compounds. So what we do when we check for some particular activity, we also check extracts and fractions. But I should say that the Western medicine is mostly focused on pure compounds. So that's why most of the research which we do is on the pure compounds rather than on the extracts and fractions. So once you have a lead compound, there are two possibilities. Basically, I will speak in a short while about this, but first I want to show you this part here is the space for the lead finding acceleration. So what we do NMR, mostly proton NMR, and we do biological assay. And we try to correlate some signals from the NMR with some particular biological activity. So the idea is, I should say that at the conditions which we use, the proton NMR takes 5 minutes. So we run NMR, we check for some particular signals. If we see them, we go to more sophisticated assay. If we don't see them, then the extract goes to the rubbish bin and we continue with the next one. So why is this so important? So just imagine that if you are in a cancer field and if you want to do anti-cancer screening, so you have different types of cancer lines and you have different time periods from 24 hours to 48, even to 72 hours. So to save time, consumables, and everything, you just run NMR, you check for some signals, you see them, you go further. If you don't see them, you continue with the next one. So this is the concept which we are trying to develop in obesity also. So as I mentioned, once you have a pure compound, which is or fraction, which is actually, fraction means a mixture of compounds, eventually with the same chemical nature, because when you do dilution, so you do in most cases you do in a gradient mode, which means that you collect the fractions with your compound, but also there could be compounds with a similar chemical nature. So you have two possibilities. One of them is to go to preclinical and clinical studies, which I should say that it happened to me only once so far, but in most cases, the compounds which we isolate are going to the rubbish bin. So this rubbish bin should be full of garbage, but in our case, it's full of natural compounds. So for the young people in the lab, as you also, they are around your generation, most of my assistants. So I should say that this is quite disappointing and frustrating. So you collect the plants, then you dry them, grind, produce extract, then you start fractionation, then you collect fractions, then you run NMR, mass spectrometry, and a number of analytical techniques to prove your structure. And then you go for the targeted biological activity. And finally, it appears that it's not active. So all this process is very lengthy and quite frustrating in the end. So I normally give one example. So do you know who this person is? So this is Hans Krebs. I'm pretty much sure you all know this person because of your lectures in biochemistry at least. So Hans Krebs was awarded with Nobel Prize in Physiology and Medicine for the citric acid cycle, or we call after him Krebs cycle. But 16 years before being awarded with the Nobel Prize, he sent his paper to Nature and it was rejected. So this is the original rejection letter in which they say that, yeah, the work is very nice, but what we always say when we don't want to have a paper. So just every time when you get a frustration from whatever paper rejected or project or so on, just keep in mind that a very nice revenge could come up afterwards. Okay, so metabolomics, just a little introduction. Metabolomics was the term metabolome was introduced something like 24 years ago. And nowadays, when we say metabolomics, we mean both qualitative and quantitative determination of all metabolites in an organism. So here we put to this definition at given conditions.
4 cubes. So we need to repeat what we did before 3 more times. 1, 2, 3.
Sorry, there is a noise which is okay, thank you.
Sorry, I don't know what's please continue.
So we put to this definition at given conditions because you should understand that the metabolome is dynamic. So what we do mostly, we take a picture of the metabolome at certain conditions, but in 1 hour, in 6 hours, at the day after, the conditions are completely different, so the metabolome is changing. And why is so interesting for us? Here I put some quotations from Nature and Science paper, which are actually highlighting the emergence of metabolomics in personalized medicine, and in particular in cancer therapy. So there are several examples which are showing how powerful metabolomics could be when they take a biopsy from the cancer patients to make a prognosis for the survival and for the way of treatment. And also there are several beautiful examples showing how metabolomics could be used to prevent. Just imagine doing metabolomics with urine, and you determine some particular compound which is increased. So then you go to the doctor say, okay, see, if you keep doing your life like this, in 1 or 2 years, you will develop diabetes. So now is the time to change. So prevention is also a very good way to cope with several important diseases, and metabolomics is contributing in the last year quite a lot on this. So for us, metabolomics is interesting because together with the transcriptomics, proteomics is forming the systems biology. Okay, so as I said, we believe metabolome term was introduced something like 20 years ago, but metabolomics is not so new. So this is a famous urine wheel, which is coming from around 16th century. So by checking the color, the smell, and the taste of the urine, at this time people were able to determine different type of disease which human already have. So I believe that this is one of the first examples for the use of metabolomics. Okay, so this is one another expression of what metabolomics is. So probably many of you have seen these big statues which are coming from Easter Island. Originally, it was thought that this is the main body of the statue, but very recently it was figured out that the biggest portion of the statue is actually laying down underground. So this is a projection, and this is how the statue looks like. So to me, this is metabolomics. It's macroscopic, unbiased approach. So if you want to see the whole picture of what you do, perhaps metabolomics is very suitable. But if you are just happy with this part, then you can stay with the traditional techniques like HPLC, either HPLC or UPLC, GC, and so on. Even TLC, thin layer chromatography, which we all do in the labs, it could give you quite a valuable information if you want to see this part. But in case if you want to see the whole part of the picture, then metabolomics is eventually a very good solution into this direction. As I said, we are using NMR mostly, or I should say predominantly. We also use GCMS and UPLCMS in some particular cases. So from the NMR, as probably also Nancy told you, the most frequently used is proton NMR. So at some conditions you can use C13, but we try to avoid C13 for many reasons. So we also do two-dimensional NMR. Here are some examples. I will show you TOC, total correlation spectroscopy, and HSQC in a short while. You can even do NMR-based metabolomics in three-dimensional mode, like plotting together HSQC and TOC. So once you have your data collected, then is coming the data processing, which consists of metabolite identification and structure elucidation. This is eventually the most difficult, time-consuming, and for the young people, annoying part, because unlike the mass spectrometry, which is for most ways of doing mass spectrometry, there are databases in which you can check for what compound is eventually the one which you see. So here there are also still some databases, but mostly we rely on our in-house databases which we develop. Then is coming the data bucketing, which is I will show you how we do this, but I'm pretty much sure that Nancy also showed you similar approach, and multivariate data analysis. Okay, before generating the data is coming the sample preparation. The sample preparation is important because what you are going to see out of the NMR or mass spectrometer is what you extracted before. So we use a protocol which was developed in Leiden, and I should say it's quite simple. So we use this KISS rule, which stands for keep it safe and simple. The idea is that if you need to prepare 100 or 200 samples per day, then you need a really robust and reliable extraction process. So we mostly use a mixture of methanol and water. At some certain conditions, depending on the chemical nature of the compounds which we want to isolate, we can go to different type of solvents like chloroform, but in most cases, methanol water is quite okay for us. So as we all know, there is no solvent which is universal. So depending on what you want to see eventually, then you can choose different solvents with different polarity. But if you want to see just the whole picture, or if the plant is not studied at all, then methanol water is, from my experience, I should say it's quite suitable. Okay, so as I mentioned, data bucketing. Data bucketing is just cutting the spectra into small buckets or bins, which could be of different size. Normally, as a gold standard, we use 0.04 ppm, but this may vary depending on what you want to explore in your extract, and eventually what kind of technique you use. For instance, if you use magic angle spinning NMR, this the size of the buckets is increasing up to 0.1 ppm, which is actually quite huge, but also the signals which are generated from the magic angle spinning NMR are quite wide. So then the data from different buckets or bins which are generated is transferred into a simple Excel file and processed with the multivariate data analysis tools. So in general, I put them in two groups, unsupervised and supervised techniques. So I, from my experience, have to say that I prefer to start with unsupervised technique, like PCA stands for principal component analysis. So in this way, you see the real separation between your variables. And then depending on the hypothesis which you have, you can go to more sophisticated assays like PLS or OPLSDA, which I put here on the slide, in a way to prove or disprove the hypothesis which you developed. So, okay, obesity because I mentioned obesity at the beginning. Why obesity is so interesting for us? Obesity is a real pandemic, in excess of 2 billion people. So I should say right now on the earth, we are about 7.8 billion people. So in excess of 2 billion, which is almost 1/3 of the people on the earth are with the overweight. And this pandemic has increased 3 times over the last 40 years. Right now, ages, gender, and different socioeconomic groups are affected. And of course, right now in European Union level, we have a discussion whether obesity is a disease or not, whether it should be categorized as a disease. But in any case, obesity is a condition which is then bringing a very could bring very severe problems, and overall obesity is decreasing the quality of life. So right now, at present, there are 3 types, 3 approaches to cope with obesity or to treat obesity. So these are I summarized on the slide, and they are mostly divided based on BMI or body mass index. Body mass index is quite simple. Everyone can count his or her body mass index by dividing the weight into the square of the height in meters. So if you have body mass index between 19 and 25, let's say, you are quite okay. And eventually you don't need to listen to this lecture. But if you have above 25, then could be problematic. So between 25 and 30, which is actually the first stage, the most frequent recommendation would be to change your lifestyle. If you have a body mass index between 30 and 40, then changes in the lifestyle are eventually not enough effective. So there is a drug therapy, different drug therapy treatment lines. So I should say that still not enough drug. There are several, like Orlistat and some others. Recently, a GLP-1 inhibitor was approved, and there are big hopes for this one, but still not enough drugs available on the market. And in case the body mass index is above 40, then the bariatric surgery is eventually a solution to save your life. So as you can see, neither of these approaches is as effective as we want. So changes in the lifestyle are known with the so-called yo-yo effects, in which you decrease and then increase and decrease and increase again, which is a terrible stress for the body. For the drug therapy, as I mentioned, not that many drugs, but still some of them are causing severe adverse reactions. And of course, when we speak about bariatric surgery, we should keep in mind that there is a risk from the surgery intervention. So in other words, there is a necessity to develop safe therapy lines, as well as novel drugs are continuously sought. Okay, so here are is a summary of research lines which could be used to cope with obesity. On this picture is how you can see how the adipocytes are actually the fat cells in our bodies. So this is how the adipocytes are formed. So it all starts from the mesenchymal precursor cells, which are under the process which we call commitment, are forming pre-adipocytes, which are then differentiating and forming the mature adipocyte. So here there are several key players in formation of adipocytes, like transcriptional factor PPR gamma, CBP alpha. This is the glucose transporter, and also some proteins like adiponectin and leptin. So every year or at a certain point, some cells, some major adipocyte cells are going under process which is called dedifferentiation, which is quite interesting. And here are the research lines which are used to study obesity. So adipogenesis is actually the formation of fat into adipocyte. So this is when we become fat, our adipocytes are doing mostly the process of adipogenesis. And as a result of this, the adipocyte is growing in size. So lipolysis is just release of this fat out of the cells. So if one of the approaches to find something which has anti-obesity activity is to induce lipolysis. This fat, which is already accumulated in the adipocytes, to be released out of the cells. This is very interesting. VIT is white adipose tissue. This is what all adults are having. But browning is very interesting because at the children age, we are receiving an excessive amount of calories, but we are not becoming fat because most of this fat which is formed is brown, which is then easily used for the energy metabolism. So one of the approaches which we are exploring is whether we can, at the adult stage, whether we can make this white adipose tissue to turn into brown and to be metabolized very easily. I should say that this is quite challenging because somehow when we grow up, we lose this eventually genetic program to do the browning, to change white adipose tissue into brown. Of course, obesity is related with inflammation, and this inflammation, when it's, as I mentioned, when at the beginning, this inflammation is healing the body, but when it becomes chronic, at certain point could be dangerous because it can bring cancer. Okay, and as I mentioned, the differentiation of the adipocytes, like going through this process back, is quite interesting approach to cope with obesity. So we were invited already 2 years back in 2020 to write a review for pharmacological research, and we hypothesized that this NRF2 transcriptional factor, which is quite interesting and how to say its use for the cytoprotection, could be taking part in coping with the obesity. Okay, so this is the approach which we use in my lab. So those of you who are already doing research on plants or in natural compounds, you probably already figure out that searching for a bioactive molecule from plants is like searching for a needle in a haystack. So you can spend your entire, I don't want to disappoint you, but you can spend your entire career in finding something which is there, which you believe is there, but you cannot find it. So in a way to find a solution, or not to look for a needle in a whole haystack, we decided to step on the ethnopharmacology knowledge. So my people went to some small places in Bulgaria, and they asked the mostly old ladies for the recipes which they used to treat obesity. So based on this, we selected several recipes, several plant species, which we then process in my lab. So as I mentioned, we produce extract, we isolate pure compounds, we do NMR metabolomics with this idea to try to correlate some signals from the NMR spectra with the anti-obesity activity. And for the screening in the lab, we use SGBS adipocytes. SGBS is standing for it's standing for Simpson-Guabi-Bemel syndrome, which is a very terrible, but fortunately rare genetic disease in which individuals who are suffering, they tend to grow in size and to develop cancer. So we use HGBS cells, which we treat with the extracts and the pure compounds, and we do qPCR, RT-qPCR, and Western blotting. So here are some examples of papers which were published recently. So this is Ziziphus jujuba. I think this plant is quite popular also in Indonesia, I guess. Also, recently, one of my PhD students published on Nononi spinosa and Alchemilla monticola. So for the sake of time, I will show you the data from the Ziziphus. But if you are interested in this domain, so you can eventually get back to me and I can show you, I can send you some papers. I mean these two papers which I mentioned. Okay, so here is the data from the Ziziphus jujuba. So we did molecular docking studies, and based on the targets which we have, apigenin, betulinic, and musclinic acid popped up. So here you can see how the adipocytes looks like. These are non-differentiated. This is the vehicle, and this is the situation when they were treated with the crude extract. So see the situation when the extract, the extract concentration is increasing from 5 to 25. And in particular, you can see the situation with the betulinic acid, which appeared the most active one with respect to the inhibition of fat accumulation within the adipocytes. And here at the bottom is the stimulation of the adipolysis, which as I mentioned, is the release of the fat. So in this case, we didn't figure some significant activity, but in case of accumulation of fat, betulinic acid appeared quite effective. At the next stage, we checked some key players in energy metabolism and mature adipocyte development, like PPR gamma, which I mentioned, this transcriptional factor CBP alpha, and ACC stands for acetyl coenzyme A carboxylase. This is on the top is the data from the RT-qPCR, and at the bottom is the data from the Western blot analysis. And in all cases, we figure out that betulinic acid appeared actually quite active. So we also hypothesized that the activity of Ziziphus jujuba extract and its active molecules might go through the PI3K ACT signaling pathway. This pathway, I don't have too much time to go into details, but this pathway is taking part in the energy metabolism, but it's also very important for different types of cancers. So again, we figure out that betulinic acid appeared the most active, and to cut the long story short, here you can see what is the eventual mechanism of action, what's going on. We hypothesize that the activity of the extract, which is abbreviated like here, as you can see, APG is apigenin and BA is betulinic acid. So it goes through the insulin receptor, which is interrupting the PI3K ACT signaling pathway. As a result, decreased expression of CBP alpha, PPR gamma, and CRBP. And in opposite way, when this pathway are not interrupted, we observed increased lipid accumulation, acetyl coenzyme A carboxylase, and adiponectin. As I mentioned, we are primarily working on pure compounds. This one example, rosmarinic acid, a very beautiful compound. It is a phenolic compound, which is formed by caffeic acid and 3,4-dehydroxy phenylactic acid. This is a dimer. So we checked whether rosmarinic acid could help us to cope with different types of obesity, and we figured out that its activity is going through TGF-beta, interleukin 17A, and CBP alpha pathway. So here is a summary of what's going on. So it's increasing the expression of FEL10 and 17A while decreasing TGF-beta, and inhibiting the adipogenic genes at transcriptional and translational level. So as I mentioned, adipogenesis is the formation of the fat into the cell. So if we can prevent this, then eventually the cells are not going to become fat. So this is very detailed. That's why I'm just showing you a little example. But if you feel like you need more information, do not hesitate to get in touch, and I will show you. I will send you this paper which we published in Food and Chemical Toxicology. At the next stage, we decided to combine two natural compounds. So this is a combination of caffeic acid and chlorogenic acid. Chlorogenic caffeic acid is, as I showed you at previous picture, is half rosmarinic acid, let's say like this. And chlorogenic acid is coming from the green tea. So probably in Indonesia, you use quite a lot of green tea. A green tea is promoted for its eventual health benefits in coping with obesity. At least in Europe, they say like that. So we combine caffeic and chlorogenic acid, and here you can see on this part when both of them were used at concentration of 50 micromoles, 50 micromoles caffeic acid and 50 micromoles chlorogenic acid, we observe a very strong release of the fat into the media. So here is the eventual, the hypothetic pathway, how hypothetic explanation how the activity occurs. We believe it is through AMPK and through the activation of some browning genes like PGC1 and E1 alpha and UCP1. So again, this data was published. So if you feel like reading the whole paper, just do not hesitate to let me know. So the next stage, all of the data which I showed you is the cell culture level. So cell culture level is very handy and powerful, let's say like this, when you want to do a screening, because just imagine that you have 100 compounds or 100 samples which you want to screen. So eventually you cannot go to mice or to whatever animals, because this will be very costly and take quite a lot of time. So cell cultures, we always start with cell cultures. We filter the compounds or the samples, and we eventually end up with one or two, and then we go to organism level. So the organism level which we use is a C. elegans. These are nematodes. They look like this, and here you can see how they are moving in when we grow them in my lab. So why these nematodes are interesting? So their genome is sequenced, and there is a relatively high homology with the humans, and C. elegans is a very nice model if you want to study neurodegenerative diseases, obesity, aging, and longevity. So another domain of research in my lab besides obesity is longevity. So we use C. elegans. So this is when we make them, we feed them with the glucose, we make them, let's say, fat, and then we treat with our compounds. Okay, so as I mentioned, besides obesity, we are interested in longevity. Longevity is interesting because what is already known is that the population are getting older. This is pretty much clear. So here you can see the projection of the WHO by 2050, what is expected to happen with the population. So here dark blue are the countries where 30% or more than 30% of the population will be at 60 years old or older. So with the white blue is percentage between 10 and 30. So Indonesia is somewhere here. So you are not that much affected. by this projection, but still more than many countries in Africa, for instance. So in a linear range, this is our life. So we have different stages, but what is important is that with the aging, our health is eventually changing. So there are several diseases, which some of them you can see here on the slide, which are related to aging. And our hypothesis is that health, in general health, including anti-obesity, anti-obesity activity, or less obese lifestyle and longevity can be improved by slowing aging. And if some of you think that this is a sort of fiction, I would like just to remind that 100 years ago or 120 years ago, the average lifespan of the humans was 50 years. So just only for 100 years, 120, because we are already 2022, we managed to increase the lifespan by 60%. So just imagine our continuous effort, what will happen with the average lifespan if we can increase with another 60. Okay, so as I mentioned, we are doing also quite a lot of biotech, so I don't have too much time to go into details, and I'm not sure whether it will be interesting for everyone, so I just want to show you how we use NMR, because this is actually, the idea is the metabolomics. So we use, in my lab, we generate so-called hairy roots, which are in vitro cultures of the plants, which are actually like a copy in the nature. In nature, every day when some susceptible plants are infected with the soil bacteria like Agrobacterium rhizogenes or tumefaciens, they are developing a so-called an infectious disease. In case of tumefaciens is Crohn gall, in case of rhizogenes is hairy root. So we copy this phenomenon in the lab. We then grow these hairy roots in different cultivation vessels, in some bioreactors, and the bottom point is that they are active producers of bioactive molecules. So these are some hairy roots which are overexpressing GFP, but here I show you one example of the work. I hosted a student from Barcelona, from University of Barcelona, who came, this is Diego, who came to my lab. In this particular cases, we wanted to find another way to utilize tobacco. So you know that Nicotiana tobacco is used to smoke its leaves, but due to the banning efforts of most of the regulators in the world to reduce the smoking, so growers of tobacco are having difficult time. So in this particular case, we wanted to engineer the biosynthesis to the stilbenoids like resveratrol, which is already having quite beneficial properties. So in this case, we overexpress some genes from the stilbenoids bioproduction, and we try to inhibit by using RNA silencing some competitive pathways like the biosynthesis of flavonoids. And here you can see this is the NMR. By plotting the data, we can easily, this is the principal component analysis, which I, as I mentioned at the beginning, is unsupervised, and it's really giving the real separation. So just by simple plotting the metabolome of transgenic and non-transgenic plants, you can easily group them in two groups. You can easily distinguish between transgenic and non-transgenic plants. Then you can see what are eventually the differences. And here is the map of which type of compounds are going up and which are inhibited eventually. So again, this was published some time ago. We also did overexpression of some bacterial halogenase genes. Here is the metabolome, NMR again. This is the Zoom aromatic region, mother plant in green, and transgenic hairy roots in red. So this idea to overexpress bacterial halogenase, it's a very long story, but to cut it, it was published a couple of years ago in Nature that when you overexpress bacterial halogenase in Catharanthus ruseus, then you can eventually get halogenated secondary metabolites. And halogenation is believed to increase the biological activity. So our hypothesis was, okay, we will express some bacterial halogenases and we will check whether the secondary metabolites which we are interested in are having halide. So by using NMR, you can easily see what's going on with the whole metabolome, as I mentioned. So when we did so, we figured out that there are signals here like this, which are absent in the mother plant. So we were quite happy. And then we did HSQC. And we figured out that it's a very simple molecule, unfortunately, but still it is NMR is showing a very great potential into this direction. So this is a set of papers which we published in the past 2 years for the work on Mycobacterium tuberculosis. This is a bacteria which is used to, in this particular strain is very similar to the virulent one, and you can check your compounds for the anti-tubercular activity. So in this particular case, we use usnic acid, which is not from plants origin, it's coming from moss, but you can see what's going on with the metabolome and the correlation between metabolome and some key genes and transcriptional factors in bacteria. Okay, so all of the data which I showed you so far is from the proton NMR, but for the exact identification, you need two-dimensional NMR. So this is not a picture of 2D NMR, it's a picture of a conference which was taken in Hawaii some years ago. Can you find me on this picture? So it's more or less the same with the proton NMR. So if you are enough lucky, you can do an assignment of your signals to some of the signals from your spectra to some particular compound. But in most cases, the spectra looks like this. And in particular, in the sugars region, you need quite a lot of imagination to be able to find some specific signals. For this, we use 2D NMR. This is TOXI, total correlation spectroscopy. This is how TOXI looks like. And here at the bottom is the Zoom aromatic region. So in this particular case, we wanted to distinguish between verbascoside and martinuside. These are phenylethanoids, but what is interesting here is that the difference between these two compounds are the methoxy groups here and here. And as probably Nancy told you, under the conditions which we use, the signals of the methoxy groups are at 3.8, which is actually the sugars region, so you cannot identify them. But in case when you use TOXI, you can see a very beautiful signals of both compounds, and you can distinguish between very close structures. We use also NMR for the quality control. This is Rhodiola rosea, very beautiful plant, which has a huge market in Europe. It is known to have adaptogenic properties. And in particular, in the north part of Europe, due to the less sunlight, when you feel depressed, the first line of treatment is to get some Rhodiola rosea products. And because the market is huge, it's somehow dirty. So what we did, we bought, here you can see only 2 examples, but we bought everything which is available on the European market. And here on the label, I don't know whether you can see well, but I can read for you. It says that there is a specific ratio between certain compounds. So this is the structure of salidroside, and this is how a commercial product look like. So we also did the HSQC, very beautiful signals of salidroside and no rosa veins. So then we started checking around and we figured out that most of the products are adulterated with rhodiola crenulata. And rhodiola crenulata is the Chinese rhodiola. So if you go to China, Chinese pharmacopoeia is saying that the products on rhodiola should be from rhodiola crenulata. But in Europe, in the Western world, in United States as well, so these products are from rhodiola rosea. So by using NMR, you can easily distinguish between real and fake products or adulterated products. So we cannot say whether it's intentional or not, but still this is a sort of adulteration. So one more example is Sambucus. Sambucus is a very nice plant, and there are several products in Europe, which here on the slide you can see Sambucol, which is used, as you can see, for cold and flu relief, and even there is a Sambucol for kids. But at certain conditions, these plants accumulate cyanogenic glycosides, like this structure from the slide you can see. This is sambunigrin. Sambunigrin itself is not toxic, but under certain conditions in human body, due to the hydrolysis, it can become toxic. So the presence of this compound is not desired in your product. So this is again the NMR spectra of the green fruits at the bottom. Then in the middle are the mature fruits and the leaves. So in this case, we didn't find sambunigrin in any example, any sample of the wild plant material, neither in the products. But according to the literature, in some certain cases, especially when the fruits are still not mature, you can detect sambunigrin. So normally when I speak about metabolomics and NMR, people are asking me whether you can, by in the crude extract, whether you can identify new compounds. Of course, the answer is no. But when you work with some specific group of compounds, you know where the characteristic signals are. And when you run NMR and when you go to the spectra, you figure out in some particular cases that there are some signals which you haven't seen before. So then you go to the classical isolation structure elucidation, while here in the case, we run NMR at the beginning, and then we figure, oh, at the iridoids region, there are signals which we haven't seen before. So then we went to the isolation, and we managed to isolate these two new compounds for the first time. So in this case, you can use NMR, as I mentioned, for a macroscopic approach. You check what's in your extract, and then if you see some signals which are eventually you haven't seen before, then you go to the isolation and structure elucidation. Okay, so it's already 1 hour. And I should go back, no, not go back, but I should end up with my talk. So I hope that I managed to convince you that NMR is quite suitable platform for accelerated lead finding, eventually in drug discovery. So it is very powerful to understand the mode of action of the healing herbs. So there is no doubt that we have many beautiful plants which could be used for medicinal purposes. As I mentioned, still 4 out of 5 people on the earth are using plants for their primary healthcare needs. But we need to know how they work, what could be eventually the side effects, and when we buy them in a form of product, to make sure that this is a real product and it's not adulterated. And as I mentioned when I spoke about what metabolomics is that right now we take a picture. We can determine a number of compounds. If you combine NMR with the mass spectrometry, you can extend the range of the compounds which you can identify in your sample. It could be plant, it could be food, it could be microbes, it could be human cells, just you name it. But the metabolome is dynamic. In 1 hour, in 6 hours, and the day after, it's completely different. So we need to move from a movie, from a picture to the real movie. So one of the solutions, if we speak for NMR, is the magic angle spin in NMR. We are pushing on this, but still the signals are quite wide, and eventually this is the future when we have a better magnets in our hands. Yeah, I can say like this. Okay, so these are the people from my teams and my collaborators which are helping, and this is my team. As I mentioned, the center at the beginning, so this is the home page, and we also have a Facebook page, so feel free to follow us or to get back in touch if you want some more information on the subject domains which I presented to you. And at the bottom, I cannot read this, but I believe it says thank you for your attention.
Wow, great. Thank you for that last word. Ya, terima kasih atas perhatian Anda. Okay. Thank you, Prof. Milen. Wow, it's so amazing talks. Yeah, I forced myself just to focus on your presentation. Okay, thank you. If I can highlight some points from your talks that Prof. Milen mentioned that plants are important source for pharmaceutical relevant compounds. I think everyone here agree with that. Yeah, he gave example about vincristine, isolated from Catharanthus. The plant is also plenty in Indonesia, and currently that compound currently used for cancer treatment. However, this plant resource also has some challenging issue. First is about the complexity, and also these plant species can be in a danger because of over harvesting, because we want to get the compounds from it. And during his talk, Prof. Milen gave an example of the research done in his lab. First, he successfully identified some compounds important to fight obesity. Milen, sorry. So actually, during the pandemic, I previously was slightly overweight, but then after the pandemic, I work from home, and then I become obesity type class 1. And then I understand what you say, like yo-yo effect. I exercise a lot, I lost few kilo, but then I become lazy and I regain it again. So I understand very well about the yo-yo effect. Unfortunately, I have to explain that. Okay, this is very interesting because Prof. Milen isolated the compounds by using NMR metabolomics, and he used a very interesting technique, which is focused on the adipogenesis inhibition and also the browning of white adipose tissue in human. This is very interesting technique. For that, he already isolated many compounds, like what I can remember is rosmarinic acid and muscarinic acid, and I think the other one is betulinic acid, and some other compound. And then he also gave example about how metabolomics can solve the issue about the danger faced by this kind of important plant species. He used metabolomics also to study about the differences between the metabolome of mother plant and also with the cell culture, and also the what you do is transgenic plants, and also you use NMR metabolomics for quality control of some important herbal medicines. Okay, so I think we already have some questions. We will move to the next session for the question and answer. Before I read the question in the chat room, maybe is there somebody who want to ask directly in this forum? Ada yang mau bertanya langsung? Is there anyone who want to ask directly to Prof. Milen? If you hesitate with the English, you can say in Indonesia and I can translate it for you. Ada yang bertanya?
Sorry, I want to ask.
Okay. Thank you, Prof. Hanifah. Prof. Milen, this is my colleague, Prof. Hanifah. She wants to ask a question. Please, Ibu Hanifah.
Yeah. I'm sorry to join just a few minutes ago.
Ah, that's okay.
because something before. I like to know about HRMAS NMR you mentioned, because I just heard this technique. HR means high resolution, right? But MAS, I just heard about the MAS linked to NMR. What is the difference between MAS and MR with the normal or regular NMR?
Yeah, well, yeah, thank you for your question. Yeah, so MS means magic angle spinning, so it is a sort of solid NMR. We call it solid NMR. It's not fully solid, but the idea is that when you use liquid samples, we need to produce extracts. And when we produce extracts, we more or less generate artifacts, because this is not the native stage in which the cells, the plant cells or animal or whatever cells are. Okay, so if we speak about bioactivities, it is pretty much sure that we would take them in a way like extract or tincture or whatever. So this is okay. But if you want to check at the native stage of the plant, then you eventually might like to avoid extraction. And if you want to avoid extraction, then there are several possibilities. You can do mass spectrometry without extraction, or you can go to the magic angle spinning NMR, which is almost without doing the extraction. So it is another head of the NMR, which is for the solid samples. You need to have this head, but I can still say that we need to progress on this quite a lot, because the signals are not the same like when you use the liquid samples, and they are very wide. And to manage to identify a compound, especially from the complex mixtures like extracts, because you know that when we produce extracts, we have perhaps 100, 200, 300 compounds inside. So this is a very complex mixture. So for this magic angle spinning NMR is still the technique which we need to push further. You know, many people are using mass spectrometry. We just got a mass spectrometer with a RAMS device, which is allowing us without extraction to check for some particular compound. So I would say mass spectrometry is progressing much faster than NMR in this direction. But with the better magnets, we will have better possibility to do magic angle spinning NMR. Or solid NMR, let's say like this.
Okay, thank you very much. Sorry, for further questions, I'd like to know about the sample preparation. Just homogenize the sample, or you just make a cut of the plants? I like to know. Thank you.
Yeah, so it depends what kind of sample you have and what kind of question you want to answer with the metabolomics which you do. So
with MAS and MR, with solid and MR, how to prepare, how to apply, or how to
okay. Yeah. In this case, I think
how
the
to
best,
put your sample in the instrument. Thank you.
Just taking the leaf, you know. So just take the leaf and put in a
just make a cut, a little cut.
Yes.
Exactly.
How to make it? This is like homogeneous, or maybe like the same condition for each part of the plants. How to make it?
Well, it depends what kind of. Then I would say that, well, if you for the magic angle spinning NMR, the solution is to see particular cases, to see what is the native stage, you know, because as I mentioned, extraction means you do some artifacts. But if you really want to check whatever compound and so, then magic angle spinning is not so good solution because the resolution is very low.
Yeah. Maybe for example, leaf, if we cut at the tip and in the middle, maybe different.
Yeah, sure, of course, even different different sections of the leaf will have different metabolome because, you know, different parts of the leaf, they accumulate different compounds. For instance, cannabis is accumulating more of the THC in the trichomes. You know, you have the leaf and you have trichomes, and most of the tetrahydrocannabinoids are in the trichomes. The same situation with the Catharanthus roseus, which is very nice plant and used to isolate these dimeric indole-alkaloids. Different parts of the leaf accumulate different precursors of these compounds, which are then put together to form the dimeric indole-alkaloids.
Okay, thank you very much. Thank you. It's clear.
Okay.
Thank you very much, Prof. Milen.
Thank you, Prof. Hanifah. Okay, so we have also some question in the chat room. I will read it for you, Prof. Milen. This is from Melanie. Does the harvest period of the plant affect the metabolomic content? If yes, which period has the best content of metabolome content? Is there any correlation such as the country, for example, plant in Bulgaria with the plant from Southeast Asia have different content because of soil or rainfall differences?
Yeah, this is absolutely true. So if you harvest the plant from different parts of the world, they have different metabolites, which we call signatures, like fingerprints. So if we take a fingerprint of Nancy and me, it will be completely different. The same with the plants, even though they are coming from the same species. So that's why metabolomics is very interesting, because this example which I showed you with the Rhodiola rosea, it is known that the best Rhodiola rosea is growing in Siberia, and they have different fingerprints than the Rhodiola which is growing in China, you know. And perhaps in Indonesia you have some species which are the best when they are grown in Indonesia, but when I buy them in Europe, they can put coming from Indonesia, and I don't know this. But just comparing the plants which are coming from Indonesia and these which are claimed, for instance, coming from Indonesia, you can by the fingerprints which are specific metabolites, you can distinguish between them. But as I mentioned, the metabolome is very dynamic, so it is changing all of the time. Not only at the harvest period, it's changing during the day, depending on the light, depending on the UV light, depending on the dark. So the harvest period, I don't think you need to use metabolomics for the harvest period. So it depends what type of plant you grow and what kind of compounds you want to isolate, you know. So even though when you use the plants as an extract or a tincture, you still have a group of compounds which are markers, either for the biological activity or for the quality of the extract. So if you follow these markers, you can say, okay, this plant is better to harvest in May or in June or in July. And still, it also depends what type of, what part of the plant you use. If you use the above ground parts, then eventually you need to harvest them before the sun is very strong, like in summer. But if you harvest the roots, then eventually you need to go even to September, when the roots are full of these compounds. So the harvest period depends on what you want to get out of this plant afterwards.
Yeah. Okay, thank you. I think it's clear, yeah, Ibu Melanie. The next question is, this is from Professor Purwantiningsih. This is also my colleague, Prof. Milen. Some lecturers also joined this webinar, so there are some questions from them as well. So not only students. She asked about the differences between screening by using metabolomics and in silico to get lead compounds for finding anti-obesity. So I think it's quite different, but okay, you can explain.
Yeah, so this is a very good question. We do both. We do metabolomics and we do in silico. So I can compare them. So in silico screening is suitable when you speak for a pure compound. So you have rosmarinic acid or vinblastine, which we mentioned at the beginning, and you have a clear target. So in this way, you can check the binding, where the binding site is, whether this binding is reversible. And how you can eventually, whether you should proceed or not. But, or to open a little brackets, that's what many people used when they screen natural compounds for anti-COVID activity. So once we had the virus genome reveal, we can check for some specific enzymes or some specific parts of the virus and some activities by in silico of natural compounds which are already known. This is quite okay when we speak for pure compounds. But when we speak for extract, which contains eventually 100 or even more compounds, then metabolomics is a sort of solution or a way, as I mentioned, for the macroscopic approach. You still don't know what could be the exact compound responsible for the activity, but just imagine if you have a good correlation between some parts of the NMR spectra with, let's say, anti-obesity activity. So instead of growing adipocyte cells, we grow them for 10 days until we have an answer. NMR, proton NMR takes 5 minutes. You run, you see, oh, I have these signals. I need to go to the adipocytes experiment. You don't see these signals, you throw out the extract, and then you go with the next one. So this is actually the point. So you have two different approaches and two different answers. Of course, the final one should be whether your compound or extract is having, let's say, anti-obesity activity. But before reaching this, there are two approaches, with the crude extract and then with the pure compounds which are responsible for the activity.
Yeah, clear ya, Bu Ati ya. Okay, so thank you for the answer. Oh, maybe I can also add something. Actually, Bu Ati, with metabolomics, we still do a lot of lab work, yeah, but in silico, I think it's fully computerized. It's also one of the difference. Okay, so here we also have another question from Ibu Melanie. She asked about the 2D or 3D NMR. When was the right time to use one of the method? And what was the significant difference between these two methods? Is it the 2D less better than 2D, or those two methods complete each other in terms of metabolomic research?
Yeah, thank you. This is very good question. So,
yeah.
Proton NMR is what we all do, but and it is quite powerful for metabolomics, because when we do metabolomics, it means that we don't care of particular compound. We do metabolomics either plants or C. elegans, which I showed you, or human cells or cancer cells or whatever. We want to check different conditions. This is what metabolomics actually is. But when we find, so just imagine we have one group of people who are suffering a disease and one group of people who are healthy. And when we compare the metabolome of both of them, we find some signals which are overexpressed in the disease people. So we want to say, okay, what kind of compounds are these? And can we use them as a marker for this disease? So just imagine that you work with urine. So you see that people suffering from whatever disease, you just name it, are expressing some signals. Then you say, okay, I can use these signals as a marker for this disease. You go to doctor, you have your metabolome done, and then the doctor says, okay, you have this possibility to develop diabetes, change your lifestyle or whatever. So then is coming the second stage. You need to identify these differences between the groups. And for the identification, eventually proton NMR, neither C13 is okay. You need to do two-dimensional NMR. So two-dimensional NMR is strong, but still depends on the chemical nature of the compounds. So for the example which I showed you with the Toxi and with the HSQC, it was easy to distinguish between these two groups. But in some particular cases, when you have glycosides or some compounds which are not that polar, then you can eventually need to go one step further. And this one step further is to try to combine HSQC with Toxi, because if you just recall what the X and the Y axis for both are, so then you have the similar axis, and then when you put them together, you can go one step further for the identification. This is the point. When you do 2D NMR and even 3D NMR, you want to go to identification. This is the real advantage of the NMR, because as I mentioned, we use mass spectrometry. Probably many of you also use mass spectrometry. Mass spectrometry is quite sensitive, but when it comes to the identification of the molecules, I can tell you, I'm in the editorial board of phytochemistry. With the mass spectrometry, you can never get your paper published, because this is by, in a theoretical way, it's not a full identification. So 2D NMR and 3D NMR, when you use them in a complex mixtures like extracts, you can go one step further towards identification of what you have. As I said, you just run two group of samples, and you find some signals which you say, okay, these 2 signals or 3 or whatever number of signals can be marker for disease, or can be marker for quality of the product, or can be marker for halal food. So I know Nancy is doing quite a lot of research on this. So you need your food to be prepared in a particular way. And if you just imagine that you have 1 or 2 signals from the samples, halal and not halal. So you have 2 markers for the halal food which are absent in the other. So you want to explain what these 2 markers are behind, because you see signals, but you still need to explain what the compounds are behind. And if you can find some characteristic signals in the halal food, then you can run. So you buy food from the market, they say it's halal. So then you run in your NMR, you see, yeah, it is or it is not.
Okay, thank you. It's a clear answer, yeah. I think because of the time limitation, I will only read one more questions. This is a very important question from Iza, yeah. I don't know, this is a girl or, okay, I think this is a girl, yeah. So Iza asked about the sample preparation. Yeah, she wants to know whether if we dry our sample first, right, using a freeze dry, and then make it a powder, is there any differences, yeah, compared to the method that you just explained, Prof. Milen? Because in your lab, I think you dry it first, right, by using liquid nitrogen, and then you crush it into a powder and then dry it into a freeze dry. But it's too expensive here to use liquid nitrogen. I think that's why she asked.
Yeah, well, it depends on the question. If you have a plant species which are used traditionally in a way to produce a tea or whatever, then this is not so important. But if you work on plant physiology or in plant molecular biology and you want to explain some very particular mechanism of action, then you need to stop all biochemical processes. Because when you cut the leaf, you still need, or stem or whatever, or if you take out the plant from the soil, you still need to keep in mind that biochemical processes keep occurring in the plant. So if you want to determine the metabolome at this stage, you need to stop them. And the best way to stop them is to get rid of the water. So what we do, we cut the leaf. So you imagine we have a plant, and then we have a pot of liquid nitrogen. We cut the leaf goes to the liquid nitrogen. Then we take it out and freeze dry it. Yes, Nancy is absolutely right. This is expensive. This is true. But in this way, we can claim that this is the situation with the metabolome when the leaf was harvested. If you harvest the leaf and if you keep it in your dryer, or if you keep it in the fridge for several days, then all of the processes which happened, biochemical processes which happened after cutting, till you analyze, these are artifacts. This is not what is explaining your hypothesis when you started. And just imagine when you take some vegetables from the market, when you forget them in the fridge for several days, what happened afterwards? These are all biochemical processes which occurred afterwards.
Sorry, Prof. Milen. Sorry, Bu Nancy.
Yeah, please.
By this explanation, I'd like to ask further about the cut of the plants for MAS and MR. Is it a frozen cut or just cut like that? Is it a frozen cut using like the tetragen cut?
Yeah, well,
or...
yeah. Well, in this case, the best would be if you cut it and immediately run for the NMR, because if you don't do this, then artifacts are produced again. You know.
But when cutting, is it in a frozen condition or not?
No. It's not in a frozen. You just cut it and you put in a cuvette, and then you went to the NMR.
Okay, thank you very much.
Okay, so actually we still have some more questions, but because the time is up, yeah, it's already quite late. I have to, sorry for other participants, I cannot read your question because of the time. Thank you, Prof. Milen. This is really interesting talk, and for sure this will broaden our knowledge here, yeah, in Indonesia, in IPB University, the student and also the researcher from IPB University, because Indonesia is one of the largest biodiversity, yeah. So I think metabolomic really a useful tool, yeah, for us to explore this excellent resource effectively and in more holistic way, yeah. But of course, there are some challenges as well, but I think we can manage to solve it by collaboration. So I wish that one day we can collaborate, yeah, Indonesia and Bulgaria, yeah, in exploring our biodiversity, also to find anti-obesity compound, because in Indonesia also we start to have this problem, yeah. We have a malnutrition problem, but in the other side we also have this kind of obesity problems. Okay, thank you very much once again, and I would like to return the time and the screen into our moderator.
Thank you very much, Ibu
Okay.
Nancy. Yeah. Thank you very much, Ibu Nancy, and also a big thank you for Professor Milen for the very amazing presentation from today. But I hope you can stay with us for a little longer, because there's a little appreciation from all of us for both of you, Professor Milen and Dr. Nancy, that will be given by the head of Formasip, Mr. Prabowo, and the head of today's webinar committee, Miss Fadila. For Mr. Prabowo and Fadila, please, time is yours.
Thank you, Beneficia. Okay. I would like to give this certificate for Prof. Milen Georgiev. My deepest gratitude. Thank you for having us for this webinar. Thank you for sharing your knowledge about metabolomics application in anti-obesity compound discovery from natural product. And I'm sure it will be useful to our future. I would like to give you this certificate appreciation as a parting gift from us. Thank you very much, Prof. Milen Georgiev.
Thank you.
I send it to your email, don't worry.
And the next certificate, I would like to thank Dr. Nancy Dewi Yuliana as moderator for this webinar, and I would like to give a small parting gift with this certificate of appreciation. And again, thank you very much, Dr. Nancy Dewi Yuliana, for the time with us. Thank you.
It's my pleasure. Thank you for organizing this event. Thank you so much.
Once again, thank you very much for Professor Milen and Dr. Nancy, and also thank you for Mr. Prabowo and Ms. Fadila. Well, ladies and gentlemen, now we come to the end of this webinar. Thank you for our speaker and also all of the participants for joining us today. I am Beneficia. Representing all of the committee, I would like to give you our deepest appreciation for your support in this event, for joining this webinar, and then we do apologize for our mistake in presenting this event. Thank you very much and have a great day today. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Milen. So see you again next time.
Thank you very much for the invitation. Have a nice day.
Have a nice day too. Bye. Pamit ya.
Thank you, Dr. Nancy.
Ya, Bu Hanifah terima kasih ya udah join. Bu Hani juga tadi ada. Aduh, saya lupa nggak welcoming beliau.